George Davey Smith, who you know all has really pioneered Mendelian randomization and done a lot for causal inference in really applied settings. And you know about gene environment equivalence as a fundamental principle of Mendelian randomization. Over to you.
Thank you. Yeah, so I'm going to talk briefly about the notion of expert knowledge in causal inference. And that's always what is said is required for the construction of DAGs, but and I'm going to discuss where that comes from. And then in the case of MR, and then the dual environment equivalence of the basis of this, and then link it to the consistency assumption more generally in causal inference. So when you see presentations of DAG-based causal inference, they often discuss about how when you're drawing your DAG, you require domain expert knowledge, all different things, background knowledge, subject matter knowledge, external knowledge, relevant theory, plausible assumptions, established scientific theories, substantive knowledge, beliefs, etc. But it always seems to have a sort of mysterious notion about, but that then allows you supposedly to construct a DAG from what you know about what you think you know about the causal world. So I'm going to discuss in the actual what is that background domain expert knowledge or Mendelian randomization. And the domain background knowledge for Mendelian randomization comes from two places. It comes from natural selection, the work of Charles Darwin, Alfred Russel Wallace, which I'm sure everyone here knows about in detail. So the natural selection is through the phenotype, which is obviously that this leads to changes in gene frequency, but it is the phenotype of the organism which has fitness, which either has greater or lesser fitness, and therefore increases in the population. It is entirely through phenotype. Genes are not selected for. There's no way a gene, the gene frequency can change the gene being selected for except through its phenotype. Now, I mean, in a sort of example of sexual selection, the Darwin most extensively about was the peacock's tail. How could this peacock's tail come about? And of course, the peacock's tail is underwritten by the DNA of the peacock, and the larger tail leads to more eggs from that peacock, and therefore leads to higher fitness. But it's the phenotype that is selected for. Or the giraffe is another classic example, the lengthening giraffe's neck through there being variation, which allow quantitative variation, which provides the necessary background for better performance, higher fitness, and therefore dissemination of the genetic variation. So natural selection is mainly on the basis of small quantitative differences, which the Mendelian characters create in continuous physiological and anatomical traits. So, I mean, luckily Charles Darwin hadn't been told what couldn't make causal claims about continuous traits because they're not well-defined interventions. So you can't do it. He just carried on and was doing it. And however, to allow the investigation of inheritance, Mendel knowingly chose traits with a qualitative phenotype. So he chose the P's with the wrinkled or non-wrinkled cases, the yellow or green color, etc., which were qualitatively different. He writes very clearly that these things are. The traits are often continuously distributed, but he wouldn't be able to identify what he did in continuous traits. In Bristol, as for Mendelian characteristics, Mendelian qualitative characteristics, we like the white tigers because Bristol Zoo have the largest collection of white tigers in the world. And they're an example of artificial selection because a white tiger in Rawa, by the Maharaja of Rawa, found a white tiger when hunting, and they deliberately crossed the white tiger with an ordinary tiger. All of the were ordinary looking tigers, but then mated the white father with his daughters, and you've got the Mendelian, and then you get the perfect Mendelian presentation. So Mendel, in terms of randomness, Mendel had two notions of chance. Firstly, was just sampling variation, the chance that you get if you do an experiment, as he did, but purely because the numbers aren't large enough to give precise answers. And 150 years before, Richard Peto clearly said about the fact that all the previous studies had been no good because the numbers weren't any good. That, as in Peter's classic initial meta-analyses in the 1970s, when that was exactly the situation. The randomized trials were too small to come up with an answer on their own. Mendel was certainly ahead of his time in recognizing this, and said you could only get these true ratios if you took huge sample sizes, and that's what Mendel did with his p-crosses. Darwin did lots of experiments. People don't know about Darwin's experimentation, often in his garden, which didn't allow for massive sample sizes. He got a rather beautiful 3-to-1 ratio with a primrose cross that he did, which is now well known. Now it's shown later to be a Mendelian trait, but his numbers weren't large enough for him to make anything of the ratios in the sea. He was just doing massively too small studies. Didn't notice the regularities that, of course, Mendel did. But then Mendel, that's mere chance. It's uninteresting. You can just deal with it by increasing the sample size. But much more interesting is the pure chance of the purely of what happens during the passing down of the heritable material. So he says that it's purely by the law of probability that on average, as many cases of each pollen form, capital A and small a, will unite equally. So it's a chance activity. That's the segregation, and then the independent assortment is that the different traits based from different loci also segregate independently through these two levels of randomization. Now the second one, the independent assortment, of course, is limited to the extent that there is some linkage through things that are related on chromosomes. But it's a remarkable biological process that produces the randomness, and it is the magic and method of meiosis that does this, that leads to this random distribution. And meiosis provides the null model for population genetics, Hardy-Weinberg equilibrium, Mendelian expectations are the null. There's the null hypothesis and genetic analyses, and then Markovian processes versus the law of ancestral heredity, which was Galton's view of sort of purely blending heritability, was that the particular nature of the transmission meant that you had a Markov process. It didn't matter what the history of the genetic variant you get is from before your parents. That's absolutely irrelevant, unlike in Galton's calculations. And in fact, hidden Markov models were developed by Haldane before Markov wrote about Markov models, but they're now they'd be retrospectively called hidden Markov models. Allows for using genes for estimating heritability, but most importantly, and the reason that you're here, that complex life exists, that we're not just still slime mold, is that it is meiosis which guarantees that there cannot be systematic genetic cheating. Because if you didn't have two-step meiosis, be very simple. If any two chromosomes lined up, just to kill the locus next to you, and you would be instantly disseminated throughout the population. I mean, this form is genetic cheating. In other forms, life forms exist, but complex life is only allowed through that. So it's not just for Mendelian randomization. Mendel's randomness is why there is complex life. And it provides the variation for selection and fair tests, because it is this perfect randomization, which means that each time that's done, that means that each gene can be tested against each allele tested against the other allele, with everything else randomized. So it allows for natural selection, and it was a major input into the development of statistics. R.A. Fisher, for example, who obviously worked on genetic data, he just says, genetics is indeed in a peculiarly favoured condition in that providence has shielded the geneticist from many of the difficulties of a reliably controlled comparison. Even if they wanted to introduce bias, he couldn't do so because the biology is what lies above that. And indeed, the notion of the factorial trial, which comes from the independent assortment, Fisher says it's not accidental, but factorial method experimentation now have lively concerns in many fields, and models the simultaneous inheritance of Mendelian factors. And his daughter said that this is where she thinks factorial trials came from. The other foundational evolutionary thinker and mathematician was Sewell Wright, who introduced path analysis in the biography of Wright, who died at 98. Jim Crow said that Wright read his father's maths books and learned to extract cube roots before entering school, a skill that he said brought in instant lasting unpopularity with the other students, which I'm sure some people will recognise. And in his first path analysis that was accompanied by a path diagram, he had done work which a bit before this. He was looking at the coat colours of guinea pigs, and he just likes chance. It's chance which leads to which gametes come back. I mean, this is clearly randomization. And his path analysis, with his path analysis, which allowed him to dissect coat colour in guinea pigs, he says that the method depends on the combination of knowledge with degrees of correlation among the variables in a system with such knowledge as may be possessed of their causal relations. And the knowledge of the causal relations entirely came from the genetic anchor, came from the fact that you have a gene, there can only be one direction of causation from the gene. And in a debate with Niles, Wright says, why path analysis is meaningful in the genetic context is because of the concrete phenomenal actuality of the results was not provided by analysis. Actually drawing the diagram and doing all that, the maths and whatever, is trivial. What is required is actually having a firm basis that anchors and directs the way that the causation must flow. So it came from the Mendelian. The rules of Mendelian heredity. That's what allowed you to make such causal claims. And so Wright's path analyses were almost but with a couple of exceptions, were genetically anchored. He did some lovely work on assortative mating being through phenotype. Just like natural selection is through phenotype, assortative mating doesn't occur because two partners come up and they say, I like your genes. They occur, well, it might be the genes you're wearing, but certainly it's not your genes inside you because they can't tell them. And one of my favourite MR analyses is showing that in UK biobank, it is indeed assortative mating by alcohol consumption is indeed because of the alcohol, in the alcohol consumption. there's only a couple of exceptions. He did one analysis with antenatal environment and said that there couldn't be reverse causation there. And his IV, one of his IV analyses that he did, he and his father also developed some sort of variables, him and his father, Philip, right, together. That was the one. And other statisticians recognise this. This is Egon Pearson and Kirksey Neyman in correspondence, and Pearson said, by 1929, I'd come down firmly to agree with Fisher that phi distributions should not be used, except in the case where they were based on real knowledge, e.g. in some Mendelian problems. So, even people who didn't normally think that way would agree that genetics David anchoring, I'll skip it is too snide. Well, quite funny. And then, of course, path analysis was then widely adopted from the late 1960s, 1970s, outside of genetics. And there's the statistician, Louis Gutman, just said, genetics is, he's taking the piss here, by the way. Genetics has got one modest framework for paths. In contrast, you read everything outside of genetics, social epidemiology, any other forms of epidemiology, whatever now. And they draw these graphs without any anchors, and that he was saying that the very weak foundation that they rest on. Okay, so but non-anchored DAGs are obviously predominating causal inference today. So back to MR. So Mendelian randomization uses the special properties of germline genetic variants, which is that that nothing causes them, the arrows must go from them, and how they influence one phenotype to allow the phenotypic causation, i.e. how one phenotype influences a downstream phenotype, to be analysed. And it was related strictly to Mendelian principles, i.e. that there is randomization. Now, the randomization does occur between between siblings. When they've got the same parents, then that is literal randomization. I've been at meetings where people will stand up and say that Mendelian randomization is neither Mendelian nor is it randomization. When I say people, I mean the icons of the causal inference field have said that, say this. And if they could actually show that, then they should get a Nobel Prize. Because if you could actually do some work which shows that the Mendelian rules are not true, then you deserve a Nobel Prize. But they are true, by the way. So the idea that Mendelian randomization is not randomization is ridiculous. It is randomization, and between of the genes of the genetic variants, and between siblings, that is random. So what we said 28 years ago was that is what Mendelian randomization is, but there were no data. There were no large-scale studies of siblings. You couldn't look at siblings. That's developed. We showed it as that, and so we referred to the populations. It was approximate Mendelian randomization, because when you're not looking at between siblings, if there are population ancestry differences, they can generate population stratification, which is one of the violations of MR. And we said that the moment that's the best we could do, and it should be called approximate. So we should have used the term approximate. We should have carried on using the word approximate MR. AMI, the antimicrobial resistance, didn't exist then, so we could have got that abbreviation. And Mendelian randomization is not, well, in my view, not always or merely IV analysis, because the special properties of germline genome is their randomized nature. It is not that they are necessarily work as an instrumental variable in terms of proxying for exposure, although those are now the most often how it's most often implemented. It's that the perturbation that they generate is randomized, and that allows you to do some really quite clever things. And Nicola Cherry did lovely work on sheep dip exposure not through using the genetic variant related to sheep dip, although if sheep dip use was a variable in UK Biobank, there was 20 papers where people GWAS using sheep dip and claimed to have shown causal effects. Now, what they did was they looked at a well characterized biological variation in peroxidase, which is a detoxification of what's thought to be the agent, the part of sheep dip, which caused fatigue and symptoms in the people who worked with it. And there's a lot of claims for compensation, etc., for using the health consequences of sheep dip use. And they did this lovely study. Just all you had to look at is the exposed only study. So I really definitely said case only design, but exposed only studies is a great design when you have a randomized perturbator. So in people who use sheep dip, who just look at people, all of them that use sheep dip, that genetic variant related to adverse consequences. It did not relate in any way whatsoever to sheep dip use. IV assumption 1 is clearly falsified, is not an IV. But it did tell you that the sheep dip, it told you that sheep dip related to these outcomes. There's lots of other examples. So many of the first few papers, many of the examples were not using an IV. An example of Mendelian randomization within IV, a sort of quite an important early example because it stopped the pharmaceutical industry developing inhibitors of C-reactive protein, was related to C-reactive protein and coronary heart disease. Many studies had shown that C-reactive protein, which is an acute phase reactant, anything bad happens to you, your C-reactive protein goes up, you get an infection, you smoke, you get overweight, all these things relate to higher CRP. You get very strong association, statistically very strong association, which attenuates about 1/3 on statistically adjusting for a wide range of confounders. This is a sample of the very wide range of confounders that were adjusted for in a collaborative analysis. And of course, as I say, everything bad, my big old, that's really bad. And a bit of a smoker, and having high blood pressure, having high BMI, everything bad goes with higher CRP. Everything good goes with lower CRP. Not surprisingly showing association with coronary heart disease, but every one of these everything that was measured was adjusted for, but you still saw a strong association in the population. But the genetic variant, MR does, when you have sensible genetic variants, MR does do exactly what it says on the tin. You have a genetic variant in the cis region of the CRP gene, the gene producing CRP, the promoter region variant, strongly related to CRP level, but not related to any of the confounders. So by dividing people up by their genotype, you've got two groups which had, on average, very robust difference in CRP level. There were no differences in confounders, and when the MR was done, it was absolutely null. And the pharmaceutical industry stopped developing an inhibitor for C-reactive protein because they realised it was unlikely to work. And I think the most interesting thing on this is actually that is what a completely non-causal association in epidemiology looks like on adjustment for your as measured confounders. That's what it looks like. And of course, we've had many examples of this in randomized trials where you get incredibly strong associations that are robust to adjustment. And that's because the two assumptions of no measurement error in confounders and no missing confounders is if you have epidemiological data, that is always wrong. In standard epidemiological data, there will be measurement error in your confounders and you will have unmeasured confounders. But the fundamental assumption of Mendelian randomization, though, is that the genetic variable and the environmental factor you want to modify are equivalent. Now, again, there's just hundreds of examples from monogenic disorders which demonstrate that level of equivalence. This is one disease called pellagra, which we now know is caused by long-term deficiency of vitamin B3, niacin, and there's a condition known as heart-nut disease, which is a genetic disorder in the uptake of tryptophan, which is converted to niacin. And they produce the same condition. If you're a doc, the differential diagnosis is between each other. Heart-nut disease is rare. So you have this equivalence between genetic variation influencing a niacin level and the environmental insult of niacin deficiency. You're deficient in niacin for two reasons, but they produce the same downstream effect. And this is just a trivial truism in developmental genetics. Mary Jane West Everhart calls it gene-environment equivalence, gene-environment interchangeability. Sir Campbell Villett famously said, no doubt all environmental effects can be mimicked by one or several mutations. I think all environmental effects on an endogenous disease or endogenous disease processes, that is true. I don't think, for example, there's a mimic of being exposed to sheep dip, which is actually leads to getting more exposed to sheep dip, or in fact, a mimic of getting run over by a truck, but it is true for other things. And gene-environment equivalence is assumed by the breeder's equation. That's what's used in breeding for trying to estimate how much selection on the phenotypic characteristics of an organism of economic importance can improve their output, their milk yield, their meat quality, for example. And how you see gene-environment equivalence is you just take the heritability is the additive genetic variation, that's the total phenotypic variation, and you just scale. You scale your expectations from that gene-environment equivalence assumed there. So it requires justifying in all situations, relates to biological processes that are influenced by genetic variation. So if you consider BMI as the phenotype, or any adiposity marker as the phenotype, you would think that things that there might be genetic equivalence with would be like higher calorie intake. And indeed, the largest effect, common genetic variant called FTO, related to adiposity, BMI, relates to higher calorie intake. Those would be equivalent. Same might be the case for physical activity level. There's of course the example was given of Miguel Hernandez gives of, you have your leg chopped off, that would reduce your body mass index, chopping your leg off, but it's unlikely to make you more healthy. But there's not going to be a gene related to chopping your leg off. But there are genetic variations. Smoking, which is related to lower BMI, but that's not going to be equivalent to any way you would think of manipulating BMI. So you go to 2 or 3 examples of, well, the examples just of gene-environment equivalence has been worked out. Probably one of my favourite examples, many other randomization still probably, was this one, which was the only within family data the only within family data we could find anywhere in 2003 for genetic variation outcomes, which is looking at a genetic variant in the MTHFR gene, which relates to homocysteine processing, folate processing. And folate levels are really had been measured in studies there. And we found that the mothers carried 2 versions of the variant related to lower folate activity. Then the offspring had twice the risk of being born with a neural tube defect. You have a beautiful negative control because, of course, the father's genotype can't influence the in-utero environment, and the father's genotype was unrelated to tube defects, and the offsprings, showing that it's actually through the intrauterine effect of folate. But you also demonstrate the problem with acid effects, because it's the parent's genotype that matters. But when you're looking at something like the intrauterine environment, of course, the offspring, by having a weighted average, an average of its parental genotypes, the offspring genotype does relate to the outcome, but it's the intrauterine stage where this is generated, and you could learn all of that from those data. And of course, folate supplementation from the periconceptual period onwards does substantially reduce the risk of neural tube defect. And in that situation, the exposure you're interested in is maternal folate levels, the outcome neural tube defect, the genetic perturbations, this MTHFR variant, the equivalent environmental perturbations of dietary folic acid deficiency or folic acid supplementation. When the mechanistic equivalence is strong, the dose equivalence is reasonable in terms of what the folic supplementation is and the effects of the genotype, and the timing is equivalent because it's the maternal folate that matters, and the mother's genotype will influence the maternal folate whilst she's pregnant with the offspring. So in that situation, you have strong equivalence with lots of beautiful cellular data showing demonstrating that. Jack talked about HMG co-reductase. So I'll move on to slides, but there's so many in the situation. LDL cholesterol is a pin-up because there's just so many equivalent situations. So HMG co-reductase or statins, there you go. They mimic genetic variation in the gene HMG co-reductase. They are HMG co-reductase inhibitors, and through inhibiting that, they reduce LDL cholesterol levels. So your genetic perturbations, those variants, the environmental perturbations, statins, mechanistically strong equivalence. The dose equivalence is weak because the variation in HMG co-reductase produces relatively small differences, and the time of equivalence is only partial because you'll start taking the statins, and the trials for statins are started at age 40 or 50 or 60. But of course, you have the genetic variation from conception, and therefore you have your LDL cholesterol levels are different over a long period of time. So, think of the equivalence in those three terms of mechanistic dose and timing. Those give the things you want to line up. And it is an example. It's an example of where a treated-only group study could be informative. Jack talked somewhat about, I think, genetic variation related to compliance, which we'll have to think about whether compliance to statins would relate to other factors that might influence outcome. But you could have genetic variation in something which just modified the effect of statin on the outcome. And people have looked for these that didn't influence baseline LDL cholesterol. Then all you need is the group of people, the large group of people, on exactly the same dose of statin, and in cases only, in exposed only, people who are on statins, you'll be able to tell whether that had an effect on coronary heart disease. PCSK9 inhibitors, another cholesterol one, genetic variation related to smoking heaviness, doesn't relate to smoking initiation. This is the 25 varying, it's very strong mechanistic equivalence. It's a nicotine receptor, and it relates to people smoking more heavily. It doesn't relate to them starting smoking, but they smoke more heavily than they started. They inhale more, smoke further down the butt. The dose equivalence is moderate, and the time is strong. The genetic variance is only having an effect if you are a smoker and while you're a smoker. So again, you have a rather nice negative control group because you're looking at people who are never smokers. The genetic variant doesn't relate to the heavier smoking, it doesn't relate to the outcomes. So these are the sort of strongest cases for Mendelian randomization. There's lots else. There's lots of this. I've got, here now, breakfast skipping. So breakfast skipping is available in UK Biobank. It's been GWASed, and there have been so many papers. Breakfast skipping, it's really, really bad for you. Eat your breakfast. And this is a review, systematic review of all the studies, and it says 22 are classed as high evidence, consisting of systematic reviews, meta-analyses, or randomization studies. The MR, as you might imagine, suggests breakfast skipping is really bad for you, but it is ridiculous, ludicrous. This is why gene variant equivalence is particularly one reason for always considering it, because it instantly tells you whether something is ridiculous to study as an exposure. There's obviously no, you've only got 20,000 genes. you're not going to have a gene which has a very specific effect on skipping breakfast, because there's probably 20,000 more important biological processes required. So you know that you're going to be looking at just a mishmash of the genetic variation which influences things through risk-taking behavior, through phenotype or whatever. So it's completely implausible. And the larger and larger gene masses mean that you get more and more genetic variants that relate to a phenotype because of higher order processes. So we saw that beautiful pattern with CRP when you're just looking at a cis variant in CRP, not related to confounders. If you now look at the genetic score for CRP based on genome-wide significant variants, related to confounders, as you imagine, because BMI influences CRP, so all your BMI variants relate to that. Smoking influences CRP, so you just pick up these higher order factors. So to consider CRP as a phenotype, cis CRP variants would obviously have equivalence, but like BMI related variants, smoking related variants, IL-6 related variants, etc. And probably anonymous trait, anonymous trans variant wouldn't. So in the causal inference fields generally, of course, the consistency assumption, whether it's argument, whether it's a definition, assumption, axiom, or theorem, but is discussed, sometimes referred to as treatment variance irrelevance assumption. So in the genetic area, in Mendelian randomization, this would relate to genetic variants that contribute to the usual distribution of a trait in a healthy population, not through pathology or modification of exogenous insults. So, so we see it as everything bad influences your CRP. One thing which influences your CRP definitely is having atheroma, atherosclerosis, or cancer, or having many illnesses. So any genetic variants that relate to those will in turn influence your CRP. So the consistency is going to be between the variants that contribute to the usual distribution of the trait. And you can see consistency between consistent genetic variation and exogenous manipulations, that's in the HMG co-reductase or PCSK9 inhibitors and other areas. And I think there are implications of the source for identification of genetic variants here. One way of reducing the number of very spurious variants through developing disease, etc., is to use young populations before they've started smoking, etc. And the chopping off the leg example for BMI, there's an example that was used for the violation of the Breeder's equation, which was about cow. This is from 1966, eating swallowing some wire and then getting ill through that. But that's not going to be reflecting anything for which there's genes could be equivalent to eating wire. So the violation is sort of obvious when you know what you're looking for. So Mendelian randomization is based on experiments. Natural selection is the outcome of a vast number of randomized trials produced by the Mendelian laws. That randomization is what allows natural selection to work. And natural selection is actually a subcategory of Mendelian randomization, with fitness being a phenotype under investigation. Fitness is a phenotype like any other. And in principle, you can see natural selection as a multivariable MR problem. When you aim to identify and selected for trait and correlated traits, because one of the issues in looking at natural selection is to actually work out exactly what is the trait which is being selected for. And that realization opens up a parallel large literature in the evolutionary biology space, which could well inform something. Obviously ran out of inspiration. I'm going to stop now. I've only got two more PowerPoints. But anyway, that literature could be very valuable to actually go back to in informing how MR works. And people in these fields, Mendelian randomization in artificial selection and natural selection, It's been used in things that are economically important, like milk, like cows, and like rice. And now, if you want to read more about Mendelian randomization, there's Alan's primer. Or if you actually want a very good book to read, I mean, it's a good read, but it's not a book. Then there's Bill Provian's book on civil right and evolution of biology is a masterpiece. Thank you.
Thank you very much. Question, comment? Maybe I'll start then. So how do you establish equivalence, gene environment equivalence?
Yeah, so it's got to be established through biological reasoning. And so, I mean, I could go back to the case of folate. I mean, you establish it actually by looking at, you can then go back and look at cellular processes and how the genetic variant influences processes within cells, and how folate supplementation influences that variation. So it's got to be established. Yeah, it's established through biological reasoning. So that's that's your that's your expert side of the knowledge that comes into it. But the genes are giving you randomized evidence on that,
yeah,
which you can then test against and look at it in such studies. And those sorts of studies are being increasingly done with, especially with single cell type data, etc.
But what, so maybe people who investigate breakfast skipping will see some gene environment equivalent where you don't.
No, well, not they did any cellular studies. They wouldn't. Sorry, that they wouldn't. So and in the end, it's yeah. But
yes, please.
Just wanted to go back to possibly throwaway remark about genetic variation not being a well-defined intervention, because
no, it's not genetic variation not being a well-defined intervention. I'm saying that people say that intervening on traits like studying cholesterol or HDL cholesterol. I had many discussions with many of these people about. They don't see it as something that you can study. It's just a continuous trait. We don't look at continuous traits. You can only study it if you have something that you say. If you have the well-defined intervention. So if you say that genetic variation is a well-defined intervention, then fine, but it means you can consider continuous traits, because that is how natural selection works. So if you don't believe in natural selection, then it's fine sort of saying you can't think of traits as having a causal effects. But if you actually believe in evolution, then traits can have causal downstream effects in fitness. But back to your question, Jonathan. Sorry, I didn't.
we puzzled me further, which is a common experience for them, but just taking the obvious example of statins and cholesterol, everyone, there's no dispute now what statins is bad.
Yeah.
Cholesterol, a continuous trait, is probably related to plants. I'm just unclear what you.
Well, that's. I mean, I'm not sure, but some people wouldn't think you can make the inference to cholesterol. You can only make the inference to a well-defined intervention. I've certainly had those discussions with people who make those, who say, the point that you can't look at BMI. You can't make causal claims about adiposity. So I think you can. That's what I'm saying. I think natural selection is a rather beautiful demonstration that you can.
Well, we've already said, and probably we can agree that we may well believe that the causal effect of reducing BMI through exercise isn't going to be the same as the causal effect of reducing BMI by the same amount.
By cutting off a leg, yeah.
So, we agree.
Yeah, well, sounds like it. Let's have a drink. Let's celebrate later. Finally, we've agreed on something.
Thanks for the talk. Do you think the classification of evidence in medicine, this sort of hierarchy of evidence, has done more harm or good overall?
Yeah, that's a interesting question. Probably, I think that there was a time when there was large amounts of things were done or believed without randomized evidence from observation. I mean, I'm probably the only person old enough to, live through this period. Jonathan, almost, but he wasn't. He's not an epidemiologist. 2 years younger than his office, were a very large number of dietary factors, which these observational studies with results which looked precisely like the CRP result. But that's why the claims were made on those bases, that you had these robust observational associations which were attenuated a bit by adjustment. And then the randomized trials, one after the other, massive randomized trials costing hundreds of millions, failed to support that, and things were done which were harmful, like giving up babies high dose oxygen, whatever. I think that when you can do randomized controlled trials, you want to do randomized controlled trials, and I think one needs to. I think it's a really interesting point about when can you use observational evidence without trials, because it has been wrong. Observational data have been wrong so many times. And observational data are wrong because the no measurement error, no missing confounders applies, and that applies to methods like target trials as well, as much as it does to other methods. So, I think the relaxation of drug licensing, which the FDA are pushing through. I mean, JFK, really old. The other Kennedy, RFK Jr. Is he? On the one hand, they say they're slagging off the pharmaceutical industry, etc. On the other hand, they're being their handmaidens by allowing a lot of real world evidence into license. So, do I believe in the pyramid of evidence? No. And I do think, because I think you should. Kate's going to talk about triangulation. I think you should triangulate. Even trials have been wrong as well. I mean, and I don't know if you've got press outside of the UK, but the Cochrane review on the anti-amyloid drugs, I mean, it's an appalling systematic review, appalling meta-analysis, and trials can get the wrong answers and things change, it's thinking of ways of which of updating knowledge. I do think that, and I think here Denison will probably agree with Jonathan about it, one of those rare things would be that once you've actually established a mechanistic causation, then other methods can be really useful for showing who that applies to. So, for example, you can't do statin trials in every single subgroup of the population, every ancestry group, every age combination with comorbidities or whatever, but you can, with now the massive genetic studies, show that you can show the HMG CoA genotype differences across virtually every imaginable population. So you can then use other methods for for transportability. I think that's the case. But I'm old-fashioned believer in that randomization, experimental randomization is a good thing.
Any questions or comments? Otherwise, a great book. Yeah, Robert? Yeah,
Thanks for the nice talk. Do people worry much about dependent data in settings where good siblings or in your observation study and then their genes are correlated and how much that affects the analysis? If you assume IID.
Yeah, so yeah, you worry about you. Well, you can use them in different ways. If you just recruit sibling pairs, then that's incredibly powerful because you look between the sibling pairs, and the genetic difference between the sibling pairs won't suffer from population stratification, isn't as damaged by assortative mating and many other things. If you but people do worry about ancestry over similarities, etc. And there are a lot of methods, especially for when analysing genome-wide association studies, for excluding relateds. So that is often done. Yeah, you exclude relateds. In fact, a study like UK Biobank is rather lovely because you can take 40,000 sibling pairs out, and you GWAS the rest of and you exclude the relateds in the rest of the group. There is 108 people in Biobank who have the same father, which is because of sperm derivation, almost certainly. Unless Genghis Khan happened to be an UK Biobank participant, yeah, so but then if you take the sibling pairs out of UKB, you have these independent tests, and also you have the between sibling test and the sib pairs and this population of approximate MR and the others, and what you actually see, and that's been quite well systematically examined now, is that, as you might imagine, you see differences. You tend to see differences of things which are, socially patterns, behaviours, etc. Much more than you don't tend to see differences in. You don't see differences in the MR answers on cholesterol between the SID pairs and the overall population, but you do for things that are more likely to relate to ancestry differences and dynastic effects that I was discussing with the MTHFR.
Okay, thank you again.